We all know that plants use light energy from the sun for photosynthesis, but have you ever wondered how they always seem to know where the sunlight is? Even without eyes or a brain, plants can bend, turn, and stretch toward the light. This fascinating movement is called phototropism, the directional growth of a plant in response to light. And here in our experiment, we focus on this exact behavior to explore how plants respond to light from different directions. By observing the growth patterns, we aim to uncover how hormones, cells, and light receptors interact to guide this movement. Understanding this process helps reveal how plants adapt to their surroundings and maximize their ability to capture sunlight. Phototropism, which literally means lightening, allows plants to position the leaves for optimal sunlight exposure. When light shines on one side of the plant, cells on the shaded side grow faster than those on the illuminated side, causing the stem to bend toward the light. This growth response ensures the plant can absorb as much light as possible to power photosynthesis and fuel development. The key players behind this behavior are hormones known as auxins, which regulate plant growth. The main auxin in most plants, indole-3-acetic acid, is produced in the apical meristem, a region made up of meristematic cells found at the tip of stem and shoots, in which they are responsible for synthesizing and secreting auxin, using vesicle-mediated transport to release it into the surrounding tissue. Because they are actively dividing and undifferentiated, meristematic cells play a critical role in maintaining growth direction and sensitivity to environmental signals.
But how do plants know which side should grow faster? It is when light hits one side of the stem, specialized light-detecting proteins called phototrophins sense a difference in brightness. These photoreceptors activate a signaling process that redistributes auxin toward the sided shade of the plant. The result is an uneven concentration of auxin, with more accumulating on the darker side and less on the other. This imbalance is what drives the bending motion. Auxin activates protein pumps in the cell membrane of the shaded side, lowering the pH of the cell wall, triggering enzymes called expansins, which loosen the cell wall structure and allows the cells to stretch. As the shaded cell elongate more on those on the light-exposed side, the plant gradually bends toward the light, a process known as positive phototropism. So when you see a plant leaning towards a sunny window, you're witnessing a remarkable display of coordination at the cellular level. The applicable cells at the tip sense light detection, phototropisms relay the message, and auxins adjust growth accordingly, providing that even without a brain or nervous system, plants can sense and respond intelligently to their environment.
To investigate phototropism in a controlled setting, we designed an experiment to observe how plants respond to light from different directions. Our main research question was, how does the direction of light influence the growth orientation of plants? Based on what we know about auxin distribution and cell elongation, our null hypothesis predicted that if plants are exposed to light coming from one direction, then there will be no significant difference in the direction of their growth, and they'll grow vertically regardless of the light source. On the other hand, our alternative hypothesis stated that if plants are exposed to light coming from one direction, then they'll bend and grow toward the light source because of the uneven auxin distribution that promotes cell elongation on the shaded side.
To test this, we use Vigna radiata, also known as mung bean seeds, as our model plants due to their fast germination rate and clear growth curvature. A total of 90 seeds were used and divided evenly into 3 groups of 30. Each group was placed in shallow plastic trays, each measuring 20 by 15 by 3 centimeters, lined with 3 layers of damp paper towels to ensure consistent moisture. To preserve humidity, each tray was lightly covered with plastic traps punctured with holes for ventilation. Each group was exposed to identical full-spectrum LED lamps placed 20 centimeters away from the seedlings. The only difference between the groups was the distance of the light. Group 1 is the control group, receiving the light from above. Group 2 receiving the light 180 degrees from the left, and Group 3 receiving the light 180 degrees from the right. All other environmental conditions were kept constant to ensure fairness. The room temperature was kept at 24 plus or minus 1 degrees Celsius, and each tray received 15 milliliters of water at the start, followed by 5 milliliters sprayed every 24 hours to maintain equal hydration. All trays were placed on the same level surface inside a temperature-controlled room and surrounded by black cardboard barriers to block stray light. And over the course of 7 days, during which we recorded the angle and direction of stem growth for each seedling every 24 hours. All the data was logged carefully and later visualized using Data Classroom, which allowed us to track trends, compare treatments, and apply statistical analysis to determine whether differences between groups were significant. This setup allowed us to observe in real time how directional light influences plant growth and to evaluate whether plants show a consistent, measurable response to their environment.
After completing the experiment and collecting all our data on the curvature of the mung bean seedlings, we ran a one-way ANOVA test to see if the direction of light had a significant effect on how much the plants bent. The ANOVA compares the means of the three groups, light from above, light from the left, and light from the right, to check if at least one of them is statistically different. Our results showed an F statistic of 520 and a p-value less than 0.01. This p-value means there's less than a 1% chance that our results happen randomly, so we can be extremely confident that the direction of light really did cause differences in growth curvature. Then, to figure out exactly which groups were different, we ran a post hoc Tukey test. This test compared each pair of groups. The results showed that every single pair had a p-value below 0.01. That means all three groups, the control with the light from above, the left light, and the right light, were significantly different from each other. Specifically, plants exposed to light from the side, either left or right, showed a much greater bending compared to the control group where the light came from directly above. The biggest difference was between the left and right light groups, showing how clear the plants grew toward their light source. These results support our alternative hypothesis, which predicted that changing the direction of light would significantly affect the curvature of the seedlings. The data also connects to how phototropism works. Auxin hormones move towards the shaded side of the plant, causing those cells to elongate more and making the plant bend toward the light. This response helps the plant maximize light absorption for photosynthesis, which is essential for its growth and survival.
Our experiment on phototropism connects really well to the ideas from the Radiolab podcast, Smarty Plants. In that episode, the host talks about how plants seem to know things like where light is or even when they're being touched. Even though they don't have brains or nervous systems, they explore the idea that plants can sense, process, and respond to their environment in surprisingly intelligent ways. In our case, we saw this same kind of behavior firsthand. The mung bean seedlings in our experiment didn't just grow randomly. They sensed where the light is coming from and bent exactly toward it. That shows how plants are capable of processing information about their surroundings using hormones and photoreceptors instead of neurons. It's like their own biological communication system, just slower and chemical instead of electrical. Our results, especially how each group's curvature changed depending on light direction, clearly support from the podcast called Plant Awareness.
The second podcast, From Tree to Shining Tree, dives into something a little different but still deeply connected: how trees communicate underground through networks of fungi called mycorrhizae. The podcast explains how this network allows trees to share nutrients and signals, almost like an internet for plants. Even though our experiment focused on above-ground responses to light, both studies highlight how plants are incredibly interconnected and responsive to their environment. While our mung beans were talking to the light, trees in the podcast were talking to each other through underground fungal networks. Both show that plants have complex systems for sensing and reacting, whether through auxin redistribution in phototropism or through chemical signaling across roots. So, connecting both podcasts to our experiment, we can see that plants, though rooted and silent, constantly interact with their surroundings in purposeful and coordinated ways. Whether they're bending toward the light or sharing resources through soil networks, they demonstrate forms of intelligence that don't require a brain, just adaptation, chemistry, and communication.
In conclusion, our experiment showed that phototropism allows plants to actively sense and respond to light through auxin distribution and differential cell growth. The mung bean seedlings bent predictably toward the light, demonstrating purposeful and measurable behavior. By observing this response, we gained insight on how plants interact with their environment, using chemical and cellular mechanisms to adapt and survive. This stresses that even simple plants aren't passive. They have intricate strategies to thrive, revealing the sophistication of plant behavior in nature.